Okay, now I want to talk a little bit about the experimental design that they had for this particular paper. What Stoeger et al. Did was they acquired a particular mouse strain from the National Institutes of Health, and they sacrificed different cohorts of mice across 5 different ages, from 4 months, which is a very young mouse, to 24 months, which is on the older side in terms of mice. And with each of these sacrificed mice, they separated the tissues and then generated RNA sequencing with each of those tissues. The whole idea of this investigation was then to compare one particular cohort, one particular age of mice against the youngest, which was the 4-month-old. So for each particular tissue, like in this example in the brain, we have some gene A, which had an expression difference between the 9-month and the 4-month-old mice of around -10. That's just an example. And this is a normal experiment. This has been done many times before, and the findings have been relatively minimal. But their hypothesis is actually different. They actually believe that aging might act in a global fashion, which is much harder to identify from a common differential expression analysis. So what they did for this experiment was actually quite different. Instead of looking at the difference in expression between these sort of different group-aged mice, they decided to take each of these genes and characterize them with a number of features. So here I have an example of different features that might explain a gene, such as transcript length, whether there's some particular transcription factor that's regulating that gene, or a different microRNA that might be regulating that gene. So they've identified just over 2,200 features for each gene that were procured from the literature, and I'll include a link of that below. And they basically have this metadata for all of the genes in all of their differential expression analysis. So instead of doing differential expression, they wanted to use these features to try and see if they could actually predict that differential expression. So what they did it sounds a little bit similar to a logistic regression, but they used a gradient boost algorithm, and I will include a link to a video explaining what the algorithm does, but it's very similar to a linear regression, except they observed that in some cases this approach can have a lot better specificity. And what's cool about this approach is they use it to basically predict that expression. You can get some idea of what the actual expression was, and then from that approach, which uses this tree sort of decision tree process with some machine learning that I'm not going to explain right now, they're able to identify which of those features are actually the most informative and the most important in terms of predicting the expression difference between those different aged mice. So here I've given an example where feature 1 was ranked 200 out of those 2,000 or so, where feature 2 was ranked 2nd. So it was pretty high up in that ranking. And with that approach, they could start to identify what features of genes are actually having the biggest impact on their change in expression with age.